Hey everybody, welcome back to another Anatomy and Physiology live stream today. Today we are really focusing on cell biology and histology for A and P. How's everyone doing? Happy Saturday. Hello. How's everyone doing? We're going to be doing a quick live stream today, a quick 60 questions for anatomy and physiology. We have a bunch of questions on cellular biology, and we also have questions on histology, and then a little mix of other things today. But how's everyone doing? Happy Saturday. Thank you guys so much for being here. I'm Ollie. These are my anatomy and physiology practice review live streams. We go live multiple times a week, covering anatomy and physiology topics that you guys have questions on and things like that. I go live multiple times a week, at least 4 times a week. Probably going to be bumping that up as we start the fall semester, and I might start doing some lecture style ones as well. We're going to be doing some practice questions today. But hello everybody. You guys are awesome for being here today. Welcome. I'd appreciate if you guys would give the live stream a like. The more that you guys do like the live stream, the more that it gets it out to as many people as possible. Thank you. I appreciate it. Thank you so much for the GGs. Thank you. Thank you guys so much for being here. Like I said, the more that you guys do like and share the live, the more that we can help as many people as possible. In anatomy and physiology, it helps just push it out a little more, so I would appreciate it. It means the world to me. I love doing these live streams. I love helping people. I love making anatomy and physiology super easy and super engaging and fun for everybody. I don't think your professors are giving you enough practice, but that's what I'm here to do. I'm here to give you the practice that you guys need and want. But we'll go ahead and get started now. Now that a few people have joined in, I'll just give a little quick introduction one more time, and then we'll get started with the questions. Hi, hey, how you doing? My name is Ollie. I go live multiple times a week covering anatomy and physiology topics, giving you guys practice questions to do to make sure that you not only just pass your classes, I want you to absolutely ace and crush them as well. I have my master's degree in molecular medicine, and I've been tutoring anatomy and physiology for about 5.5 years now. All my resources and things like that, I post videos, I have tutoring. You can tap on my profile for more information on that. But without further ado, let's get started with the questions today. I'm just going to flip the camera around and we'll go ahead and get started. Thank you all so much for being here on a Saturday. Happy Saturday. Alrighty, can everyone see the questions? Alright, and then we'll go ahead and get started. As soon as I see a yes in the comments, I'll go ahead and start reading the questions off. Just a quick yes will help me out just to make sure that you guys can see. Yes, thank you. I appreciate it, guys. Thank you. Number 1, identify the organelle responsible for ATP production. So like I mentioned, we're going to have a lot of questions today. Going into some cellular biology questions is really important. I know a lot of these at least in my cellular biology notes, I made sort of a chart on organelles' functions and whatnot. It's best to maybe do some flashcards, memorizing what organelle pairs with which function. It's super important, especially when you get into a little more of the physiology and biochemistry of cells in your human body and stuff like that. But thank you guys so much for the follows as well. Yeah, we can do some cardiovascular system this upcoming week. Number 1, the answer is A. A lot of you put A. That is awesome. The mitochondria, which you guys, a lot of people, pretty much everyone knows that is the powerhouse of the cell. But essentially what mitochondria does, it has oxidative phosphorylation, which is going to generate most of the ATP. And then we'll move on to number 2, which best compares our rough ER and our smooth ER functions. And we'll go over some of the other ones, the other organelles in later questions as well. So stick around for those. But welcome in, everybody. Hello. A lot of you just joined. It's nice to meet you. You're in cardiac muscles right now. Awesome. Yeah, we'll be going over some cardiovascular and AMP2 on Tuesday. But welcome in, everybody. Hello. How's everyone doing? My name is Ollie. Welcome to my Anatomy and Physiology Practice Review live streams. We are going over Anatomy and Physiology's cellular biology chapter today. And then we're also going to be doing some histology questions as well. We're on number 2 now. Number 2 asks the rough ER versus the smooth ER functions. The answer is C. The rough ER is going to synthesize proteins, while the smooth ER is going to synthesize lipids. Number 3, what is the main function of the Golgi apparatus, A, B, C, or D? Good morning, Day. How you doing? Welcome in, everybody. Happy Saturday. I know I will be, after this live stream, I'll be getting into some of the college football today. I don't know if anyone here is a big college football guy, or a guy or gal. But welcome in, everybody. Thank you guys for 1,000 likes on the live stream already, guys. That is insane. Thank you all so much. I appreciate it. But number 3, the answer is D. It is our protein modification and packaging. Proteins from the ER are going to be modified through glycosylation, phosphorylation, and they're sorted into vesicles in the Golgi. Number 4, what is true about lysosomes versus peroxisomes? A, B, C, or D? What do you guys think for this one? Welcome, everybody. My name is Ollie. I'm going to be going live multiple times a week covering A and P for you guys, giving you guys the practice questions to help. Yep, the Goji apparatus is the post office. Yeah, I always call it the Amazon nowadays. But yeah, it's kind of like the post office, basically modifying it, packaging it, and sending it off. But welcome in, everybody. It's nice to see everybody. It's nice to meet you all. Happy Saturday. Welcome. I don't know who's in anatomy and physiology this semester, but if you are, please say hello. Please say when you start and what your if you're an AMP1 or AMP2. Thank you for the roses, Maya. Thank you. Number 4, the answer is C. Lysosomes are going to digest macromolecules. Peroxisomes detoxified hydrogen peroxide. Hydrogen peroxide is very toxic inside, and we have our peroxisomes to detoxify that in our body. Number 5, what describes the nuclear pore complex? A, B, C, or D? AMP2 currently, that's awesome. Hello, I'm in AMP1. You have 1, 2, and 3 on Tuesday already, and your histology lab is coming up. Make sure with those histology slides, you're making sure you're taking pictures of the microscope slides so you can practice labeling them. It's hard. Jinx brings me back. Awesome, Kate. Bio 24, Life with Nat. Tasha, AMP2 currently. May, AMP1. Well, awesome. Good luck, guys. You guys are going to crush it this semester. We'll be going live multiple times a week, making sure that you guys get the practice that you need to make anatomy easy for you. Number 5, the answer is B. It is the gateway for RNA. It's going to move proteins in and out of the nucleus. Moving on to the next page of questions here. Which cytoskeletal filament is correctly matched with its function? A, B, C, or D? You have microbiology this semester. Good luck in microbio. We're going over some cellular biology questions today for A and P, and then we also have some histology questions at the end. Thank you so much for the roses. I appreciate it. Thank you, Paul. Thank you so much. Number 6. We're on number 6 now if you're just following along. We're talking about which cytoskeletal filament is correctly matched with its function. The answer is D. It is microtubules is correctly paired with the function for vesicle transport and mitotic spindle formation. Microtubules are essentially going to track how our motor proteins. It's going to form our mitotic spindle as well. Number 7, which is going to best describe the fluid mosaic model? A, B, C, or D? What do you guys think? Welcome, everybody. Welcome. Today we're going over Chapter 3. We're going over cellular biology for anatomy and physiology, and then we're going to be getting in a little bit to Chapter 4 with the histology today. And then I have a mix of some other A and P1 questions. But number 7, what do you guys think for number 7? A, B, C, or D? If you're new here, you just comment A, B, C, or D. I appreciate all the likes on the live stream as well, guys. We're at 1.8 thousand likes. The more that you guys do like and share the live, the more that it just pushes it out to people. We can help as many people as possible in their anatomy and physiology exams this semester. Thank you so much, Kate, for the roses. I appreciate it. I had a little voice crack there. Number 7, the answer is C. It's dynamic it's a dynamic phospholipid bilayer with its embedded proteins. So again, the fluid mosaic model, it being a bilayer, meaning it has 2 layers. The embedded proteins are going to give it some function. Number 8, which is the difference between the passive and active transport? A, B, C, or D for this one? What is the difference? Thank you so much for the follows as well. I'm going to be doing anatomy and physiology multiple times a week here on TikTok. But welcome, everybody. It's nice to meet everybody. You guys are absolutely incredible. But welcome. Hello, everybody. Hi, Catherine. How you doing? I hope you're doing well. It's good to hear from you again. Good to see you. Number 8, the answer is B. Active transport requires energy. Passive does not. Number 9, which is an example of a receptor-mediated endocytosis? What is that going to be? A, B, C, or D? Welcome in, everybody that just joined us. Hello. It's nice to meet everybody. My name is Ollie. I go live multiple times a week covering anatomy and physiology topics. I'm here to make anatomy and physiology super easy for you all. We're covering cell bio for A and P today and histology with a little mix of some other questions for A and P at the end as well. Number 9, the answer is D. It is the uptake of LDL cholesterol. So receptor-mediated endocytosis, cells use receptors to bring in specific molecules, specifically here LDL. Engulfing bacteria is phagocytosis. Insulin secretion is exocytosis, so this is exo, and bulk fluid absorption it's called penocytosis. Number 10, what does the sodium-potassium pump do? This is super important. Super important. The sodium-potassium pump. What is this going to do? This is important for maintaining the electrochemical gradient, which is essential for your neurovascular and your muscle function. A, B, C, or D for number 10. What do you all think? Just comment your answer choice of A, B, C, or D. Awesome. Thank you guys. We're also at 2.4 thousand likes on the live already. That is insane. Hell, you've been helping me in A&P one. That's awesome, Better. Thank you so much. I appreciate it. Awesome. Number 10, the answer is C. It is going to pump 3 sodiums out. So we are going to pump 3 sodiums out with 2 potassiums in. Number 11, what is an example of a secondary active transport? A, B, C, or D for this one. What do you all think? Happy Saturday, everybody. Thank you guys so much for being here. Welcome. Thanks for all the follows as well. It's nice to meet you guys. I remember more than I thought. It's been a long time. That's awesome, Kate. No, that's awesome. I'm so glad to see that. Awesome. That makes me happy to see. Number 11, the answer is B. Is a glucose sodium symporter. So it's going to use sodium's gradient that we talked about that gets established by that sodium-potassium pump to bring glucose in. Sodium-potassium pump is a primary one. Diffusion is passive, and the calcium pump is primarily active. Primary active, I'm sorry. Number 12, which is an example of endocrine signaling? What do you guys think for that one? A, B, C, or D? Just comment your answer choice. Thanks, guys, for the follows. It's nice to meet everybody. Hello. We're also at 3.6 thousand likes already. Thank you, guys. I appreciate it. Again, the more that you guys do like and share, the more that it gets it out to more people. Keep up the great work. Wish I had you in undergrad. Thank you. Yeah, thank you as well, Kate. Have a great day as well. Enjoy your weekend. Number 12. The answer is D. When you're referring to the endocrine system, you are referring to things, hormones getting pumped down in your blood, so hormones traveling in the blood. Number 13. G protein coupled receptors act through which mechanism? I got the hiccups randomly. Every time I get the hiccups whenever I live stream, I swear. It's only whenever I live stream it happens. I'm going to take a sip of my drink. What am I eating? I actually have not eaten yet today. I have the worst morning nausea ever, and it's not till like noon or 1 where I can eat. Number 13. The answer is C. Is the activation of second messengers like CAMP. Basically, GPCRs activate G proteins. That triggers adenylate cyclase. It's going to increase one of the pathways like CAMP, DAG, IP3, etc. This is really important when you have to go over the mechanisms, especially if you go into like medical school or anytime you have to memorize pathways. Number 14, which is not a second messenger? A, B, C, or D? A, B, C, or D? What do you guys think? Number 14, the answer is ATP. ATP is not a secondary messenger. It is an energy currency. It's not a signaling molecule. DAG, IP3, and CAMP are classic second messengers. You'll learn about them a lot in pathways. Number 15, what is the role of kinases? A, B, C, or D for this one. Kinases. A, B, C, or D? Well, hopefully these questions will help make this a little bit easier for you as well. But number 15, the answer is B. They're going to add phosphate groups. Kinases are going to phosphorylate proteins. Phosphorylation means to add phosphates. Phosphatases are going to remove phosphate groups. Number 16, apoptosis is regulated by which pathway? A, B, C, or D? A, B, C, or D? Welcome in, everybody. We're going over some cellular biology, physiology today. We're on number 16 now. Apoptosis is regulated by which pathway? The answer is a caspase cascade. Apoptosis is programmed cell death, caspases such as proteases are going to cleave your target proteins. mTOR is going to regulate growth. Your MAPK is for signaling, and JAK-STAT is a gene expression. Number 17, what is the correct order of the cell cycle? We're talking about the cell cycle. What is going to be the correct order, A, B, C, or D? A, B, C, or D? What do you guys think for this one? Also, I do have all of my notes for anatomy and physiology. They're on my website. Tap on my profile for more information about that. But number 17, the answer is going to be C. It's G1 to the S phase to G2 to M. G1 is growth, S is DNA replication, G2 is the prep, and M is the mitosis. Number 18, which protein is going to regulate the G1 checkpoint? Which protein will that be? A, B, C, or D? It is P53. It is what is referred to as, I guess you could call it like the guardian of the genome. So if DNA gets damaged, P53 will halt the cycle, and it will induce apoptosis. Number 19, and remember, apoptosis was that programmed cell death. What occurs in metaphase? For number 19, we're referring to metaphase. What is going on? A, B, C, or D for this one? Welcome in, everybody. Welcome, welcome. We're at 4,000 likes on today's live. Thank you guys so much for being here. Welcome, welcome. We're at 4.6 thousand likes. I appreciate it, guys. That is insane. Number 19, metaphase is A. Chromosomes are going to align at the cell's equator. So spindle fibers attach, chromosomes line up. The nuclear enzyme dissolves earlier in prophase, and then the cytokinesis is telophase. Number 20, cyclins and CDKs function to what? A, B, C, or D for this one? How many questions do we have? We have 60 questions today. We have some cell biology, organelle functions, and cell physiology today for the first 30 questions, and then we move into some histology questions, and then I have a couple A and P1 questions. Number 20, the answer is C. They're going to control your cell cycle progression. Cyclins bind to CDKs, which is going to phosphorylate targets to drive cells through their cycle checkpoints. Number 21, what is the difference between mitosis and meiosis 1? A, B, C, or D? A, B, C, or D? What do you guys think? What is the main difference here? What is the main difference? A, B, C, or D? Yep, it will be. You can tap on my profile to get that for the replay of the live streams. I post every single replay there. Number 21, the answer is A. Homologous chromosomes separate in meiosis 1. Meiosis 1 is homologous chromosomes are going to separate, and crossing over gives you that variation. Mitosis is where the sister chromatids separate. Number 22, what is the importance of crossing over? A, B, C, or D for this one? What is the importance? I kind of just talked about that a little bit, but let's talk about where that occurs. A, B, C, or D for this one? For number 22, fantastic. It is B. It's for that genetic variation. Homologous chromosomes exchange DNA during prophase 1. That is going to increase the genetic diversity. Number 23, we have glycolysis. What is the net ATP yield of glycolysis per glucose? A, B, C, or D for number 23. What do you guys think that one might be? I'll give you guys another moment to answer. It is C. It is 2. 4 ATP gets produced, 2 is used, so the net is 2 ATPs. Number 24, what is going to best describe the citric acid cycle? A, B, C, or D for this one? What do you all think? A, B, C, or D? Awesome, awesome. Welcome, welcome. Thank you guys so much for being here today on a Saturday. I love you guys. I love doing the live streams. They're the favorite part of my day. Welcome in. We're at 4.7 thousand likes. I appreciate all the likes, guys. Number 24, the answer is B. It's going to generate NADH, FADH2, and carbon dioxide. Number 25, what is the role of NADH? So again, in oxidative phosphorylation. Thank you guys so much for the roses and the gifts. I appreciate you guys so much. Number 25, what is the role of NADH in oxidative phosphorylation? The role is A. It is an electron donor to the ETC. The ETC is our electron transport chain. Number 26, anaerobic respiration is going to produce what? Thank you guys so much for the gifts. I appreciate it. The Corgi, thank you so much. I love Corgis. They're one of my favorite dogs. Number 26, anaerobic respiration is going to produce what? The answer here is B. It is lactic acid. Without oxygen, pyruvate is going to turn into lactate in animals. Aerobic respiration is going to produce your carbon dioxide and water, and much more ATP. Number 27, beta oxidation breaks down what? Beta oxidation is going to break down what? Thank you guys so much for the follows. It's nice to meet everybody. Hello. I'm Ollie, by the way. I go live multiple times a week covering anatomy and physiology and some other topics. Today, we're going over cell biology for anatomy and physiology. We're going a little more in depth today. We did a little bit of some of the organelle functions. Today, we're kind of going over some of the molecular cell physiology and some of the biochemical pathways as well. There is biology in anatomy and physiology. Of course there is. There's biology, there's chemistry. That's what makes us up. But kind of depends on the depth. Some people go a little more in depth. Today we're going in depth. So number 27, the answer is C. It is our fatty acids. Beta oxidation breaks down fatty acids. Fatty acids break down into acetyl-CoA. They go into the TCA cycle. Number 28, the central dogma of molecular biology is what? A, B, C, or D for number 28. Just comment what you guys think the answer choice is, A, B, C, or D. But yeah, we'll be going live multiple times a week. I'm going to give you guys practice questions to make these topics super easy for you. We're going to finally make it make sense. We're kind of going to explain why questions are right and why some are wrong. And then I'm also going to start doing some lecture-style videos as well for those of you who are interested. But number 28, the answer is B. It is DNA to RNA to protein. Genetic information flows from the DNA to the transcription of that DNA to RNA, and then it gets translated, so translation into a protein. Now, speaking of this, so again, we have our transcription versus translation. 29 asks us transcription differs from translation, and that transcription what? What is it? What is the function and purpose of transcription? A, B, C, or D? What do you guys think for this one? Number 29, if you're following along. Number 29 is C. We're going to synthesize RNA from DNA. Transcription occurs in the nucleus, converting our DNA to mRNA. Translation in the cytoplasm is where we get that mRNA to a protein. Number 30, ribosomes function to what? A, B, C, or D? Last cell biology question here, and then we're going to move into some histology questions. Thank you guys so much for 5.5 thousand likes in the live already. That is insane, guys. Thank you so much for all the support today. But number 30, the answer is C. It is to synthesize proteins. Ribosomes are the site of translation, using mRNA and tRNA to build our polypeptides, forming our proteins. Number 31, we're going to move to the next page of questions here. Again, we're moving into some histology and some tissue questions for anatomy and physiology. If you would like to get the questions, all you have to do is join our TikTok sub-community. Once you join, send me a DM, let me know that you joined, and then you will get the blank test and the answer key after every single live stream. I go live multiple times a week, at least 4, so you're getting 4 practice tests a week with at least 60 questions. Number 31, which is not a characteristic of epithelial tissue? What is not a characteristic of epithelial tissue? A, B, C, or D for this one. What do you all think? A, B, C, or D? Number 31. The answer is D. It does not have a rich blood supply. Epithelial tissue is avascular, meaning there are no direct blood vessels. Nutrients must diffuse from the underlying connective tissue. Number 32. Where is simple squamous epithelium found? A, B, C, or D? What do you guys think for that one? A, B, C, or D? Where is it found? A, B, C, or D? A, B, C, or D. What do you guys think for this one? Welcome, everybody. We're doing some histology and tissues for anatomy and physiology now. We just went over some cell bio. Number 32, the answer is B. It's the alveoli of the lungs. So simple, meaning one layer. Squamous means flat. The thin, flat cells allow for the rapid diffusion of your gases. Number 33, pseudostratified columnar epithelium is going to line which structure? A, B, C, or D for this one? And the skin would be stratified squamous epithelium because, again, it's for protection. Thank you, Jordan, for the sub. Please send me a DM so I can get you these questions after the live. Again, if you join our TikTok sub community, you get the blank test after the live stream with the answer key at the end. You can go through and practice it on your own. I go live at least 4 times a week, so you'd be getting at least 4 tests a week. But welcome. Number 33, the answer is pseudostratified columnar epithelium is going to be what? The answer is going to be our trachea. D. We have ciliated pseudostratified columnar epithelium. They have what's also called, we have goblet cells. This is going to line the trachea to help move mucus. Your esophagus is going to have stratified squamous. Your stomach has simple columnar. And lungs have, again, we just talked about in the last question, your simple squamous epithelium. Number 34, we have transitional epithelium found in which part of our body? Cilia are the little projections that are going to help move mucus along the tract. But number 34, transitional epithelium is found where? A, B, C, or D? What do you guys think for this one? And if you're just joining us and you are new here, it's nice to meet you. My name is Ollie. I go live multiple times a week, covering anatomy and physiology, as well as some other topics for you guys as well. I'm here to make it easy. I'm here to give you guys practice. Your professors don't give you enough practice. I'm here to give you that practice. Number 34, the answer is B. It is the bladder. The transitional epithelium is going to allow for stretch. Think about what happens in your bladder. It fills up with urine. It allows your bladder to stretch, filling up with more urine so you can hold in your pee a little longer. Number 35, microvilli function to what? What do microvilli function to? A, B, C, or D? This is super important. Yep, no more cell bio questions. We finished those up with question 30. We're moving into some histology questions. Again, we did some cell bio earlier for chapter 3. We're doing chapter 4 now. And then I have just another mix later on in the live stream of some A&P1 questions. We try to do a little mix of everything, make sure we hit everything, just to make sure the live streams are kind of catering to the majority here and everybody gets the practice that they need. This is exactly how you get the questions. I give it to our TikTok sub community. Once you join, send me a DM. You get the blank test and the answer key so you can practice on your own. Number 35, the answer is A. It is going to increase the surface area for absorption, mostly found on your intestinal epithelial cells. Again, it increases your nutrient absorption. Cilia are going to help move. There are some there in the intestines, but it's going to help move the food throughout the digestive system, but mainly here the mucus as well there and in your respiratory tract. Number 36, which exocrine gland secretes by cell rupture? This is a bit of a tricky question. Sometimes some people have to know this one, sometimes you don't. But I decided to go over today just because somebody in a tutoring session was confused on it, so I added this question in today. They were going over it for their histology. Not many people do, but some do, so that's why I have it in here today. But number 36, which exocrine gland is going to secrete by basically cell rupture? The answer is C. It is holocrine. Holocrines are where a whole cell dies. An example of this could be in a sebaceous gland. Merocrine is by, an example of that would be exocytosis with salivary glands. Apocrine is your portion is going to pinch off like your mammary glands. And endocrine are hormones, basically getting released directly into your bloodstream. Number 37, your basement membrane contains what? A, B, C, or D? Definitely didn't know that. It's totally okay. Like I said, some people do have to know it, some people don't. It came up in a tutoring session, so that's why I kind of wanted to make sure that people knew it. But number 37, the basement membrane, the answer here is going to be B. Your basal lamina and your reticular lamina. The basement membrane is your basal lamina, your epithelium, then your reticular lamina is your connective. Reticular fibers are alone, or your elastic collagen are only would be like an incomplete description. But number 38, which of the following is not a function of a connective tissue? I'm learning. Perfect. Yeah, no, I'm so glad you're learning, but thank you for being here to learn. I really appreciate it. Welcome. We're also at 6,000 likes on the live stream today. I appreciate all the likes, guys. That is absolutely fantastic. The more that you guys do like and share the live, the more that we can help people on their upcoming anatomy and physiology exams. I know a lot of people are starting with their fall semester. But number 38, the answer is D. It is not impulse conduction. That would be your nervous tissue. Number 39, what is loose connective tissue? What is loose connective? A, B, C, or D? What do you guys think? Thank you so much for this. Thank you so much for being here. I appreciate it. Number 39. The answer is B. It's areolar. Areolar connective tissue has a loose matrix for flexible support. Number 40, elastic cartilage is found where? Where do we see elastic cartilage? A, B, C, or D? What do you guys think for that one? I also have notes for anatomy and physiology whenever I was in the class. I got an A in the class. I got 100% on my final exams. You can click on my profile for more information on that. It's in my bio. Number 40, the answer is C. It is the external ear. It's flexible. Elastic cartilage is flexible. Costal cartilage would be hyaline. Your knee meniscus it would be fibrocartilage, and your pubic symphysis would be fibrocartilage. Number 41, osteoclast function to what? A, B, C, or D? Also, I have a little bit of a mix of A and P1 questions here as well, just in case some of you are farther along. But if you're not here yet, it is perfect to help you get ahead. Thank you so much for the follows as well. It's lovely to meet everybody. My name is Ollie. I go live multiple times a week covering anatomy and physiology topics. Pretty much, in case you want to know where anything you can buy or anything or purchase, it's all available in my TikTok bio. You can tap on my profile for more. But if you would like to get the questions, I give it to our TikTok sub-community. I'm going to put up the little notification to join. Once you join, just send me a DM, and then I'll start sending you the questions. Taking this class next semester. That's awesome. Awesome. Good luck next semester. We'll be here going live. Osteoclasts are going to function to break down bone matrix. We have osteoclasts and osteoblasts that are going to build up. So we build up our bone matrix. Osteoclasts, I always think of chew with the C, or cleave. And number 42, compact bone is organized into what? A, B, C, or D? What do you guys think for this one? A, B, C, or D? Awesome. 42, compact bone is organized into B. It is osteons, or Phrygian systems. There's the structural unit of compact bone. Number 43, plasma is the what? What is plasma? What is plasma? What is the exact definition of it? A, B, C, or D? Number 43, plasma is B. It is the extracellular matrix of blood. It's basically your fluid matrix that is combined with your proteins, your ions, your nutrients. Number 44, which of the following is voluntary muscular tissue? A, B, C, or D? What do you guys think for this one? Thank you guys so much for all the likes as well. We're at 6,200. Do I have any pre-records class on your page? I don't. If you're talking about recordings, you can go to my profile. I don't know what you're really asking, but if you're talking about the recordings, just go to my TikTok bio. All the information can be found there. But number 44, the answer is C, skeletal. Skeletal muscle is voluntary. This is false. Cardiac and smooth are involuntary. Number 45, what structure shortens during contraction? What is it? What exactly is the part that is shortening during contraction? A, B, C, or D? Which part of the sarcomere, which component is the part that shortens? It is A. It is your I band. So during contraction, actin slides over myosin, your I band shortens, A band stays the same, Z lines move closer. And then the M line is going to remain consistent. Number 46, the sliding filament theory states that what? What is the sliding filament theory? You will have to know this for A and P, especially when you get to muscle physiology. Welcome everybody. Hello. It's nice to meet everybody. Awesome. Also, Jordan, if you're in the live stream still, I just saw your email. I'll respond to you. I'll respond to you right after I end the live. Thank you so much for sending me the email. Number 46. The answer is B. Myosin pulls actin filaments past each other. Myosin heads again attached to actin. They shorten the sarcomere. Number 47. Intercalated discs are unique to what? They are unique to what? Very unique. A, B, C, or D? Number 47. The answer is C. It is your cardiac muscle. Intercalated discs are your gap junctions and your desmosomes for connecting cardiac cells for a synchronized contraction. Number 48, smooth muscle contraction differs in that it what? A, B, C, or D? What do you guys think for this one? A, B, C, or D? 48, smooth muscle contraction differs in that it is B. It's going to use calmodulin instead of troponin. Calcium binds to calmodulin, which activates MLCK, which is going to phosphorylate myosin. Skeletal uses troponin. So number 49, the axon of a neuron functions to what? A, B, C, or D for number 49? So a lot of you are already answering it. Number 49, the axon of a neuron is going to function to B. It is going to transmit signals. Axons are the long extensions sending action potentials away from the soma or the cell body. The dendrites will receive it. Number 50, myelination in the CNS is by what? A, B, C, or D? Again, in the CNS. The CNS. Also, if you'd like to get these questions, you can do so by joining our TikTok subcommunity. Once you join, just send me a DM or an email with your username, and then I'll get you these questions as well. Number 50, the answer is D. It's oligodendrocytes. Again, this is in the CNS. Schwann cells are in the PNS. Number 51, which is a chemical synapse feature, A, B, C, or D? What do you guys think for that one? Number 51, what is a chemical synapse feature? I do anatomy and physiology practice tests multiple times a week. We also include some other practice tests as well, like some little bit of biochem and pharmacology and things like that. Number 51, what is a chemical synapse feature? The answer is C. It is neurotransmitter release. Chemical synapses are your vascular neurotransmitter release. Number 52, what is a glial cell? What is a glial cell? A, B, C, or D for this one? What do you all think? A, B, C, or D? What is a glial cell? The answer is B. It is an astrocyte. Astrocytes are an example of a glial cell. Glial cells are going to support neurons. So you have your astrocytes, oligodendrocytes, microglia, the epidermal cells. Neurons transmit signals. Sarcomere is for the muscle, and chondrocyte is for cartilage. Number 53, the blood-brain barrier is maintained by what? A, B, C, or D? What do you guys think? A, B, C, or D? The BBB. It's known as the BBB. Thank you guys so much for the follows as well. It's nice to meet everybody. I'm Ollie. I'll be going live multiple times a week covering anatomy and physiology topics that you guys request. Thank you guys so much for being here today. Number 53, the answer is C. It is astrocytes. Astrocytes are going to maintain that blood-brain barrier. Astrocytes are going to induce a tight junction in the CNS capillaries. Number 54, hematoxylin stains what? Hematoxylin is going to stain what? A, B, C, or D? What do you guys think? What do you guys think for this one? A, B, C, or D? Number 54, the answer is B. It's going to stain the nuclei blue or purple. Hematoxylin is a basic dye that binds to acidic DNA or RNA. Number 55, PNS stain highlights what? A, B, C, or D? Yep, we also do some microbiology a little bit here. Again, I'm including a bunch of different questions that a lot of people have been mentioning they have been struggling with. So again, we go, like I said, we go over a lot of different things. Mainly we do anatomy and physiology. We do some bio, some chem, some microbio, some pharmacology. I'm here to try to hit a little bit of everybody. And if you kind of need anything more specific for you, that's what I do offer tutoring. If you need stuff and more practice and things that's more personalized for you. Again, so I'm trying to really try to tailor to the majority of people and the requests and things like that. So again, we try to hit everything. So we did some cell bio today. Some people mentioned that they were going a little more in depth and had trouble with it. So I went in depth today. Then we did some histology, a little mix of A and P1 as well at the end, some of the other topics as well. And then we have a little bit of microbiology. Again, just trying to make sure that everybody gets a little bit of their own practice. But if you need personalized study, personalized questions, and personalized help, that's what my tutoring is for. This is free. This is here. I do this at least 4 times a week. I'm going to be ramping it up now that the fall semester is starting. But yeah, like I mentioned, I'm here doing this for free. I'm here trying to help as many people as possible. So that's why the questions are a little all over the place today. My Monday and Tuesday live streams, A&P1 specific and A&P2 specific, only A&P1 is on Mondays. And my only A&P2 lives are on Tuesdays. But number 55, the answer is C. It's going to be our carbohydrates and glycogen. So your periodic acid shift stains, polysaccharides, and glycogen magenta. Number 56, immunohistochemistry uses what? What are we doing here? A, B, C, or D? What's the function? Helpful knowledge bench. Thank you, Geraldine. Good to see you. It's good to see you again. Happy Saturday. I'm using GoodNotes. It's my favorite. Also, Notability is a good one as well. But number 56, the answer is A, antibodies to detect your proteins. Number 57, electron microscopy is going to allow for what? A, B, C, or D? What do you guys think? Also, if you would like to get these questions, I give them out to the sub-community after every single live. It's the blank test and the answer key at the end. Number 57, the answer is A. It is a higher resolution than light. Number 58, chronic smoking causes respiratory epithelium to change from what? A, B, C, or D? Last 3 questions here. We'll breeze through these. Number 58. The answer is C. So again, if you have a chronic smoking, it's going to cause your respiratory epithelium to change from ciliated columnar to stratified squamous. Number 59, Marfan syndrome involves defects in what? A, B, C, or D? If there's anything that I didn't cover that you necessarily need a little more help on, I do offer tutoring. It's available on my website. All that information can be found in my TikTok bio. I'm happy to tutor you. I'm happy to help you out. If you need a little more personalized approach, this is kind of tailoring towards the majority here. So if I can't get to everything. But number 59, the Marfan syndrome involves defects in the answer choice B, fibrillin 1. Marfan is a defect in fibrillin 1, which is your elastic fiber microfibrils, which is a little bit of your connective tissue weakness. Number 60, a pathologist diagnosing cancer often looks for what? What are they looking for? A, B, C, or D? Oh, I just realized something. I'll fix this. There's 2 of the same answer. The answer here is going to be A. It's going to be the nucleolus size. Basically, we're referring to cancer cells often have an enlarged nuclei, which is a result of an increased ribosome synthesis. Awesome. Alrighty, guys, those were all. I know. Sometimes I try to make questions fast so I can get the live stream out sometimes. Last question there. But I'm going to flip the camera around and say hello. Hello, everybody. Hello. My name is Ollie. My hair got really flat while I was looking down. But hello, everybody. Hi. Thank you for the roses. Thanks so much for being here on a Saturday. Thank you guys for being here. I really appreciate it. I'll be live again on Monday, likely at around 2:30 p.m. Eastern Standard Time. Hello, everybody. Hello. Welcome. These are my anatomy and physiology live streams where I go live multiple times a week covering anatomy and physiology. Like I mentioned, we also do some other topics as well: biochem, pharmacology, microbiome, things like that. A&P1 is a specific A&P1 day on Monday. A&P2 is on Tuesday. All the information about tutoring and everything like that can be found on my website. It's in my TikTok bio. This A&P1. We did mostly A&P1 today. We did a little more in-depth. Nice heart. Thank you. Yes, I love you guys. Seriously, I give my heart out to you guys. So this live stream, normally we do HESI and Ts on Mondays and Tuesday, A&P1, A&P2. I like to do review lectures. So yeah, we'll start doing lectures. For now, we normally just do practice questions, but I'm going to be doing lecture live streams as well. Like I mentioned, I'm a little busy with my tutoring right now, but as soon as I get some more extra time, I'm going to start doing some lecture live streams as well. But yes, so this is essentially what we do. We hop on these live streams. I give you guys practice questions because your professors, I don't think, give you enough practice. That's what I'm here to do. A&P1 is on Mondays. A&P2 is on Tuesdays. It's more tailored towards the HESI or Ts. The rest of the week is kind of a mix of what I've seen gets requested the most, what you guys struggled with the most, particularly in the A&P 1 and 2 days. And then I also mix in other questions with pharmacology, try to make sure that we kind of get a mix of everything. But again, I'm probably going to be ramping up the live streams, so hopefully we can have a little more specific tailored days. But again, like I mentioned, if you need personalized help, this is, again, we're tailoring to the majority in the live streams as well. So if there's an issue with that and anything like that, that's what the personal tutoring is for. Yeah, so if you need more personalized stuff as well. I'll stick around for a couple more questions. So on Monday, it'll be at 2:30. On Tuesday, it should be earlier. It'll probably be at 1 p.m. Eastern Standard Time. It'll be earlier. And then we should have one on Wednesday and Thursday as well. I don't have times for those yet. I'll definitely be posting about it, though. I'll make a post right before I go live. And then hopefully the following week we'll start doing more live streams. So not just the 4 times a week, but sometimes multiple times a day, more times a week, and things like that. But thank you guys so much for being here. Like I mentioned, if you want to get the questions, I give them to the TikTok sub community. It's the exact test. I guess I do have a YC. Yep. Everything is linked in my TikTok bio. You can tap on my profile for more. Oh my goodness, I had a hiccup. Excuse me. But yeah, everything is available in my TikTok bio. All the resources, all the services I offer can be found there as well. Send me a DM via Service Plus. Do I have beginner class? I've been thinking about starting those. Yeah. If you have an interest in that as well. Sorry, I'm late. I'm in California. Oh no, that's okay. Thank you for trying to be here, though, Justino. It's good to see you again. Thank you guys so much for being here today as well. If you're interested, I'm planning on doing. I'd like to do a kind of like a success roadmap into A&P 1, and also do one for. Nice to see you too again, Geraldine. If you're interested in something like that, send me a DM in the Service Plus. It's in the bottom left corner there. I'd like to do that where I kind of go over, not necessarily go in depth on A&P 1 and A&P 2 information, but kind of go over like, so this is what you're going to cover in A&P 1, and this is kind of how to go about it. This is what it focuses on. These are the important parts, like kind of the higher yield stuff, and then kind of go into like how should we go about studying that. So I'm hoping on doing some workshops like that. And some lectures like that, and the lectures are going to give you more practice, so it's going to cover information and then practice. But the live streams will probably be like a lecture-specific and then a question-specific live stream, so a little bit different. But again, it's the Service Plus button. If you're interested in something like that, let me know. I will get you set up. I'll get it set up, and then I'll send you when I do that and whatnot. But thank you guys. Have a great weekend. Seriously, I'm going to get going. Love you guys. Have a fantastic weekend. Like I mentioned, the TikTok sub community gets the questions after every live. All the notes and resources, tutoring, etc. Can be found. Tap on my profile. It's in my TikTok bio. I'll let you know what to do. And all the videos I post will be here as well. All the replays on YouTube, YT as well. They're on YouTube. That's linked in my bio, and my website is also linked in my bio. But you guys have a great rest of your weekend. Thank you so much for joining us today.